Now that you understand peptides, we're ready to talk about proteins. Remember that peptides are just simply two or more amino acids that are connected to each other. We call the connection a peptide bond or an amide bond. A protein is basically a giant peptide. A protein is biologically active, which not all peptides are. So that's one way that they're different. And by biologically active, we just mean that it has the ability to perform some sort of function in some sort of biological system. They're not all active inside humans, for example. And proteins have to contain at least 40 amino acids. 40 or more amino acids. So they have to be big. Peptides don't have a minimum number of amino acids other than 2. So peptides can be small, but proteins have to have at least 40 amino acids. Because the proteins have 40 amino acids and each amino acid has a minimum of 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 10-ish or so atoms. So each amino acid has a whole bunch of atoms, and you have to have at least 40 amino acids. So we're looking at hundreds of atoms in a protein. The shape and the structure of a protein is extremely complex, and that's just due to the sheer number of hundreds of atoms that make up the protein. So when we're talking about the shape or the structure of a protein, there are 4 different ways that we describe the shape or the structure. And the 4 different ways are increasing in the complexity of the protein. So the first one that we're going to talk about, number 1, which is the most simple way to describe the shape, is called the primary structure of the protein. The primary structure of the protein. And all proteins can be described with a primary structure or by its primary structure. And the primary structure is just simply the sequence of the amino acids the way that we were writing them for peptides. So from the N terminal to the C terminal, the order of the amino acids in the protein. And that really gives us limited information about what the shape of the protein actually looks like, but it's a good starting point. The very first thing that has to happen is that all these amino acids have to be put together in a particular order. So we're not really going to spend a lot of time talking about the primary sequence or primary structure. I know a lot of books will describe the primary structure of amino acids like they are beads on a necklace, like just sort of little amino acid molecules that are all connected to each other, something like this. So that's an okay description. But like I said, that doesn't give us a lot of information about what the shape of the protein actually looks like. So the next thing that we want to talk about is something called the secondary structure. And as far as I know, all proteins have a secondary structure as well, but I'm not a biochemist. The secondary structure is actually a shape. It's a 3-dimensional shape of a portion of a strand of amino acids. So, kind of going on this beads on a necklace analogy, if we have a protein that has all of these amino acids connected together in a primary structure, then perhaps maybe these ones right here are going to adopt a secondary structure, a unique three-dimensional shape. And maybe these ones out here are going to also adopt their own secondary structure, their own three-dimensional shape. Not all of the amino acids will participate in the secondary structure, so some of them will be left out, like these guys right here. And the secondary structures in an amino acid, there could be two, there could be more than two, and they're not necessarily affiliated or associated with each other or dictated by each other. The secondary structure of an amino acid is created when there is hydrogen bonding. So it is caused by hydrogen bonding between two amino acids that are not peptide bonded to each other. So it's hydrogen bonding between two amino acids, third try, between two amino acids that are not connected to each other by a peptide bond. The hydrogen bonding takes place, so it exists between the NH of one amino acid and the carbon-oxygen double bond of the second amino acid. And we're going to look at a picture. We're going to look at a couple pictures of it, so you'll get a good chance to see it. But the two amino acids that are hydrogen bonding are not directly connected to each other, and they are bonding through one amino acid's NH and the other amino acid's CO. When this hydrogen bonding takes place, there's two different shapes that can come up or arise. So there's two shapes of a secondary structure. And the two different shapes depend on how close these two amino acids are to each other when they start bonding together. So the two different shapes of the secondary structure are called the alpha helix. That's one shape. And the other shape, we'll leave ourselves some space, is called the beta pleated sheet. So the first thing I want to do is show you a picture of these two shapes. We're going to focus on the alpha helix first. And the alpha helix picture that I'm going to show you is from your textbook. This is figure 21.4 in your book. In my book, it's on page 730, but it depends on what edition you're using. So this is two different representations of the alpha helix. Like, one might be easier for your eyes to see than the other. And I'm going to start with the representation that's on the left-hand side. So what I want you to do initially is ignore this green ribbon that's going through this diagram, because the green ribbon is trying to help you visualize the concept of the twisted alpha helix. What I want you to do is focus instead on the ball and stick models that are inside this diagram. And I am trying to find the easiest place to start. I think the easiest place to start might be down here at the bottom of the diagram. So what we have down here at the bottom, this is a carbon atom right here. It's kind of hard to see the color of this guy because it's hidden behind the green, but that's a carbon atom. And over here in black, there's another carbon atom right there. And this in the blue, that's a nitrogen atom. And this is bonded to a hydrogen. So the hydrogens are white. This is a carbon-oxygen double bond. So in red up there, that's an oxygen. And then the yellow, you can probably tell from that, that's the R groups that make each amino acid unique. So what we have right here, we have the nitrogen to the carbon unit of an amino acid. And then because of the three-dimensional nature of this drawing, it is kind of hard to see the amino acids connected to each other, but there's our peptide bond. And what I want to do now is move over to the other diagram, because the other diagram is going to maybe make it easier for us to visualize the hydrogen bonding that's occurring between two different amino acids. So again, the same amino acid that we just focused on is right here. There it is. And this is its carbon-oxygen double bond right here. And so this particular amino acid, I really want to highlight right there, this dashed line is showing us that it's hydrogen bonding to this hydrogen right here, which is connected to, that's a nitrogen in blue. So this is right here, the NH unit of one amino acid that is hydrogen bonding, there's our H bond, to the carbon-oxygen unit of a different amino acid. And these two amino acids, as you can see, are not directly connected to each other. There's quite a few amino acids in between them. And so this pattern, this repeating pattern of hydrogen bonding between two amino acids that are not directly connected in this order creates this twisted shape. So it's like a spiral, sort of a corkscrew twisted shape, where it gets its name, the alpha helix, with helix is a twisted representation. In the alpha helix, one of the unique characteristics about the alpha helix is that the R groups of the alpha helix are always aimed outward from the helical structure. So they're always sticking out, kind of like spikes. And again, it's really hard to draw that on a piece of paper, but what it's showing is that, so if this is the alpha helix, if this is the shape of the molecule, the R groups are all jabbing out of the alpha helix on the outside surface of it. So none of them are like embedded on the inside like this. They're all sticking out. So let's go back to our notes and kind of add this information that we've got for our alpha helix. We have hydrogen bonding. We didn't count on that diagram because it's pretty hard to count individual amino acids, but the hydrogen bonding in an alpha helix is between every fourth amino acid. And this causes a twisted shape, helix, but helix is not an everyday word, so maybe corkscrew or spring would be something that's easier to visualize. Twisted, helix, corkscrew, spring shape. And all of the R groups point out from the helix like spikes. So now let's talk about the other option for a secondary structure, the beta pleated sheet. This is also out of your textbook, figure 21.5, which is on page 730 in my book. And in this, so this is also hydrogen bonding. This one's easier to look at because it's not as sitting, structures are not sitting on top of each other as much. So here we have, well, we're going to focus down here. This right here is one single amino acid unit. Here's the carbon oxygen double bond. Here's the nitrogen with the hydrogen. Here is the R group. So it's using the same color coding system where the R groups are in yellow. And then this diagram is showing the hydrogen bonding that takes place. So there's hydrogen bonding that takes place between, here's the NH of one amino acid and the carbon oxygen double bond of another amino acid. And so for this, there isn't a set number of amino acids that Like with the alpha helix, it's hydrogen bonding between every fourth amino acid. With this beta pleated sheet, there isn't a set number, but what we see are 2 amino acids that are side by side. So this amino acid, which is side by side to this amino acid, which is side by side with this amino acid, they are hydrogen bonding to a similar set of side by side amino acids. So these guys that I just circled are all side by side with each other as well. And so this creates a flat pattern of amino acids that are side by side, and due to the shape of the amino acids in general, it does give kind of a kinked up shape, which is why we call it a pleated sheet. This looks kind of like it's a little bit folded. In the beta pleated sheet, this part is a little bit hard to see from the diagram, but all of the R groups in the beta pleated sheet are either sticking up above the sheet, those are the easiest ones to see, or they are sticking down underneath the sheet, kind of like I'm finding the down ones. So if we were to look at it from the side, if we were just to kind of look at it from a side diagram, it would sort of look like this, kind of all stuck together, and all the R groups would be sticking up and sticking down, but none of them would be embedded flat within the sheet. They're all up above it or down below it, and they alternate up and down. So let's go add this stuff to our notes. The beta pleated sheet, we have hydrogen bonding, but in no specific pattern. So it is hydrogen bonding between just segments of amino acids side by side, segments of side by side amino acids. And this causes a flat shape with like kinks in it that we call the pleats. And the R groups point above and below the sheet.